Why do life-saving drug therapies work for some patients but not for others? And why does a medication that works well for one person cause adverse side effects in someone else? Some of the answers may lie in our genetic makeup. That's why researchers in the Center for Individualized Medicine at Mayo Clinic are studying pharmacogenomics to better understand how inherited genetic differences affect an individual's response to drug therapy.
So what the genome is the 3 billion letters of the genetic code that make each of us uniquely the wonderful person we are. We've never had the ability to do this before. And the Human Genome Project, which I think we can take great pride in, it was an international enterprise that involved Europe, Asia, and the United States, but led by the United States, basically set out to do what I think most of us 20 years ago thought would be impossible. I didn't think it would happen during my lifetime. That is to basically give us the outline of the sequence of the human genome. Now, everybody's outline is different because we all have about 7 million variations there that make us who we are. The Genome Project, though, the way in which it was presented to the public was as if it was a race to the finish line. No. It was a race to the starting line, because once we had that outline, we could then begin trying to figure out how to use that to help us understand diseases like cancer that we really had no ability to deal with before in any meaningful way to prevent and to treat. Number two, what made us uniquely who we are in terms of the way we respond to the medications we have.
Mayo's researchers use knowledge gained from the Human Genome Project to analyze the genomes of thousands of individuals, an enormous task, since a single human genome contains more than 3 billion DNA base pairs. Dr. Liwei Wang and a team of talented researchers use rapid sequencing tools to identify biomarkers that can predict a patient's response to drug treatments.
Now, because we know the whole genome, entire genome, and we have the technology to assess entire genome in a much faster and efficient way, so now we can incorporate all the genetic information from your entire genome instead of just looking at the individual gene. And by doing that, so we have a complete picture and how the whole genome might impact your response to a certain drug.
Analyzing these large, complex data sets requires powerful computers that can process data at unprecedented speeds.
Eventually, we will have 3 billion nucleotides coming from one individual, not even mentioning many other types of omics, like proteomics, the microbiome data. So imagine you try to integrate all of these data, and you need automatic systems, and you need very sophisticated algorithms.
To tackle this data-heavy analysis, Mayo has a strategic partnership with the University of Illinois, bringing together top thinkers in technology and medicine. Supported by funding from the National Science Foundation, this alliance is exploring better and faster ways to analyze and make sense of the pharmacogenomics team's research data. What does this work mean for Mayo Clinic patients? In the Beauty study, researchers performed DNA sequencing on breast cancer patients before and after they undergo drug therapy. Then they compare the tumor's genome from before and after therapy against a sequence of the patient's DNA in normal, non-cancerous tissue to match the genomic response to the drugs.
I think that one of the things that we learned from the Beauty study was that the cancer genome indeed is complex, and that we can identify some patients that may not derive as much benefit from standard chemotherapy.
So what we tried to do in this study is we obtained millions of data points from these patients and then try to identify patterns using novel computational methods and determine what are the novel pathways in each of the molecular subtypes, as well as in individual pathways, what are the mutations that are altered, and those mutations that can be druggable for that particular patient.
As we move forward with Beauty 2, we will be enrolling patients who have chemotherapy-resistant disease, i.e. patients that complete standard chemotherapy and still have tumor left in the breast, and making novel drugs available for those patients as part of Beauty 2 to further try to drive to improve the outcomes of women with breast cancer.
In another study, Mayo researchers are performing rapid DNA analysis of patients undergoing angioplasty procedures to help cardiologists determine the right anticoagulant drug to use after the procedure.
So Taylor PCI is so important. We are all talking about personalized medicine and how it's going to be helpful for patients. And Taylor PCI has now already become one of the largest genetic-based clinical trials in cardiovascular diseases ever conducted. So what we are doing with Taylor PCI is trying to prove that genetic testing could perhaps be helpful to identify those patients, one of the 30% of patients who could be at risk by taking Plavix or clopidogrel, and then giving them the alternative drug would improve outcomes. Less heart attacks, less strokes, less clot in the stents, etc., and less death perhaps. So that's what we are looking at.
Through the RIGHT study, the Center for Individualized Medicine is adding data on drug-gene interactions to patients' electronic medical records.
So the RIGHT study, which was the right drug at the right dose at the right time for the right person, all that stuff, the RIGHT study was really a pilot study to test the possibility that we could actually do that. We didn't know whether this would work. And we picked 1,013 local patients who had their DNA in the biobank. They enthusiastically consented to participate. And what we then did was sequence all of these genes that we currently know play a role in drug response, put that information preemptively in the electronic health record so that when their doctor writes a prescription, immediately the electronic health record, if there's going to be a problem because of their genes, sends a message to the doctor to either change the drug, lower the dose, or raise the dose, which I find astonishing.
Clearly, pharmacogenomics has the potential to revolutionize the way drugs are used to treat individual patients. With high-tech tools and a growing understanding of the human genome, Mayo researchers are helping to launch an era of truly individualized medicine, where patients can get the right drug at the right time in the right amount.